Good morning, everybody. Those here attending in person in the lecture hall, and also welcome to those attending by the YouTube channel. So, as you may know, the speaker today is Professor Irene Díaz. Thank you very much for accepting the invitation to participate with us in this cycle of seminars. Thank you. Thank you, Irene. And as it is a custom, I will say a few words about his professional career. Professor Irene Díaz Moreno is currently full professor of biochemistry and molecular biology at the Institute of Chemical Research of the Scientific Research Center Isla de la Cartuja, CIC Cartuja, in Seville. He got her PhD degree at the University of Seville, and he made a postdoctoral stage at the MRC in London, in UK, from 2006 to 2008, working on the regulatory mechanism of mRNA decay by RNA binding proteins. In 2010, she got a permanent position in the University of Seville, and in relation with her research, she is developing research projects in the biointeractomic field, as well as on the post-translational regulation of biological macromolecules. In the crosstalk, this is quite interesting, in the crosstalk between the nucleus and the mitochondria in human disease. She has served as a chair of the FEPS working group on the careers of young scientists since 2018, and is vice chair of evaluation panel for MSCA actions within the Horizon 2020 and Horizon Europe in the Life Sciences panel. So, Irene, it's your turn. Thank you very much.
Okay, thanks very much, Jose Miguel, for the nice introduction. And thanks very much for the invitation, because it's a great pleasure for me to be here in the Instituto Química Física Rocasolano, where I have really good friends for many, many years already, not only on the NMR field, but also on the crystallographic field. So, and I met Josemi a few years ago as members of the executive committee of the Spanish Biophysical Society, where I think we had a lot of fun. And also we have the privilege to share our position with Juan Carmelo, that you know, he passed away a few weeks ago, and he was a reference in biophysics for the Spanish community, and a good friend of us. So I would like to remember him. The title of the talk is about one of the favorite proteins in the group, but what I would like to invite you is to a trip through the different cellular compartments. And as an example, I will use the respiratory cytochrome C, because it's a nice example of a protein that is able to be in different cellular compartments doing different functions. So it's not only an electron transport protein involving in the mitochondria, in the electron transport reaction in the mitochondria, but also it's going to do different things in the cytoplasm, and I hope I convince you that in the nucleus is performing a new and completely unexpected function. So just to begin with, I would like to thank all the people at the biointeractomic units. This is the Guadalquivir River, and and that was in September, of course, when the weather was fantastic in Seville. And also I would like to thank the collaborators, not only in Spain, but out of Spain, because without their help, it could be impossible to perform the experiments that I'm going to present. So I will start with the canonical role of the protein in the mitochondrial respiratory chains, because it is well known that it's a soluble heme protein transferring electrons from complex 3 to complex 4 in the inner mitochondrial space. But what I would like to remark with this slide is the fact that the protein is involved in a more complex interacting network. In all the cases, the protein is involved in electron transport reactions, so these kind of complexes are very transient, the interactions are very transient, and the complexes are short-lived complexes. But still, the picture becomes even more complicated under the nitrooxidative stress, because under these conditions, the protein, the heme protein, is also able to reduce other proteins like cytochrome C peroxidase or P66. And even more complicated is the fact that under nitrooxidative stress, the protein became nitrated at the level of tyrosine. And this post-translational modification promotes or triggers the interaction between the protein and a lipid, which is typical of the inner mitochondrial membrane cardiolipid. So again, what I would like to remark that in all these interactions, electrons are move between the partners, and the lifetime of the complexes are very, very short. And actually, if we have a look at those residues that are involved in the different interactions with the different partners involving electron transfer complexes, all these residues are surrounding the heme cleft. Because they are redox complexes, so we are expecting that we need to have contact between the redox centers. But also, if we have a look other complexes in which the protein is involved, not in the context of the electron transfer, but in the context of the apoptotic signaling pathway, there are also residues that are close to the heme involving this interaction. But what this, at least for us, quite stimulating is the fact that these complexes, which are not electron transport complexes, are characterized by really long-lived lifetimes. So this is what we call transient life versus stable death. So what is new, or at least we think that we contribute in part to what is new in the field in the function of the protein in the inner mitochondrial membrane, is the fact that we can different rearrangement between the membrane complexes depending on the cellular condition. This is what is called fluid model versus the solid model. So in the fluid model, we have the membrane complexes separated each other, whereas in the solid model, we have this kind of associations in what we call supercomplexes. Like, for example, the supercomplex between complex 3 and complex 4. And this has to have an effect on the way that the electrons are transferring from cytochrome C, from complex 3 to cytochrome C, and from cytochrome C to complex 4. And indeed, a few years ago, and doing NMR, we defined not only what we call the proximal binding site, which are compatible with the electron transport reaction. It means that the distances between the redox centers are compatible to promote this electron transfer. We define other additional binding sites that we call distance. And we call distance because they were not compatible for the electron transport, but they have to, they explain a kind of meaning. And what we thought is that this distal binding site can't accumulate molecules of cytochrome C in close to complex 3 or complex 4 to enhance the electron transport reaction. So this has an impact, not only the presence of this distal binding site, but also the rearrangement of the membrane complexes in the way that the heme protein is transferring electrons between these two complexes. Because up to now, it is well accepted that there is a kind of 3D search of cytochrome C in the bulk of the inner mitochondrial space. Whereas if we have this kind of rearrangement, we can promote the electron transfer just by sliding of cytochrome C molecules on the surface of complex 3 and complex 4. And we were very, very happy because 1 year ago, the group of Peter Besinski, which is an expert in bioenergetics, defined this sliding, the possibility of the sliding of cytochrome C molecules on the surface of complex 3 and complex 4 by cryo-electron microscopy. So he was able to identify electron density on the surface of complex 3, on complex 4, but also in between. So it's possible that the 2D search for the electron transfer between the two membrane complexes is performed by cytochrome C. But still, apart from discovering this distal binding site, we decide to measure the minimal distance at which the electron transfer occurs between cytochrome C and cytochrome C1. And this was done in collaboration with a group of Pau Gorostiza in Barcelona using electrochemical tunneling spectroscopy. And to our surprise, we were able to transfer electron up to 10 nanometers, which is huge in electron transfer. So we thought, okay, I mean, it is normal that in this kind of complexes, we need to define a well-defined conformation between the two redox protein and also promote the exclusion of water molecules in the interface. So if we are able to transfer electrons up to 10 nanometers, it means that we have an ensemble of multiple conformation between the redox protein, and it means that something should be in between the two proteins. And this is what we try by molecular dynamics, and we discovered that there was an electrostatic potential through the two redox proteins, which is what we call a charged conduit. And we need to demonstrate that this is the situation. So because it was quite interesting for us that in the suppose a channel between the two proteins, the cations and anions of the salt used in the molecular dynamics were excluded. So now we are thinking that probably specific residues on the surface of cytochrome C are involved in this kind of channel. And I didn't tell you, but the protein is quite positive. It has a PI of 9.5, so it's full of lysine residues, and actually it has 18 lysine residues out of 104, so it's a lot. So we are now working in the assignment of the amide group of these lysine residues, and we were able to identify and to assign the 18 residues. And again, this is in collaboration with Barcelona, with Miquel Pons. So we are now monitoring the relaxation properties of these amine residues of this lysine in the presence of different anions, specifically in the presence of chloride. So we are doing also some molecular dynamic computational studies to see the lifetime of these chloride anions on the different lysines distributed in patches, in clusters, on the surface of cytochrome C. So we are expecting to identify if there are preferences in the way that the anions are contacting with the lysines. But again, I mean, the protein is highly regulated by post-translational modifications, and it can be nitrated and phosphorylated and also acetylated at the level of these lysine residues. But I'm going to be focused in the phosphorylation of tyrosines and how this phosphorylation can regulate the protein. And actually, I mean, all these post-translational modifications are able to modulate the well-defined functions of the protein. So the functions in the mitochondria, like for example, the electron transport function or the peroxidase activity, and the function that the protein performs out of the mitochondria in the cytosol. So for example, the function related with caspase activation. So if we focus on tyrosine, on the phosphorylation of the tyrosine, just to remind you that we used to mutate this tyrosine by a glutamate, but indeed, I mean, mimicking phosphotyrosine by replacing the tyrosine by a glutamate is not a well phosphomimetic mutant. And actually, what we did was just run some molecular dynamics on the wild type, on the phosphorylated protein, and on the mutant tyrosine 48E mutant. And as you can see, the side chain of this glutamate is flipped out of the protein, so it leaves some space to allocate water molecules close to the hinge, which is a mess because it changed completely the redox properties of the protein. So for this reason, now we are using to mimic phosphotyrosines by non-canonical amino acids, like this amino acid, paracarboxymethyl-L-phenylalanine amino acid, in which we keep the volume of the phenyl group and we add a carboxylate group at the end. And in terms of molecular dynamics, it behaves as the phosphorylated protein, and the way that we include this non-canonical amino acid is not developed in our lab, but it's used, it was took from another lab, from the Peter Schultz lab, that he developed a plasmid, a p-ball, that codifies or encodes for an unnatural amino acid tRNA synthase for a tRNA that is able to recognize the non-canonical amino acid and to include this non-canonical amino acid in the specific position that we are interested in. So what we have to do is just in the gene of the protein that we are expressing is mutated the triplet codon by a number codon and obviously changing the stop codon for our protein. So the system works, and we were able to solve the structure, the NMR structure of the phosphomimetic mutant, and the overall folding is pretty similar to the wild type. So the difference becomes from the dynamics of the molecule. So this loop that contains the mutation is more flexible, and this dynamic, we have check all the function of the protein. It has an impact not only on the cytochrome C peroxidase activity, but recently we have coming back to the electrochemical tunneling spectroscopy and measuring the distance at which the current is occurring. And we realize that we are able to modulate this distance just including some charge on the surface of the protein. So when we work with the phosphomimetic mutant and we measure the distance for the electron transport reaction between the phosphomimetic and complex 3, towards cytochrome C1, the distance was shorter, and the lifetime of the complex was significantly increased. So now we know that we are able to transfer electrons at long distance. We can modulate this distance, but still we need to identify this charge conduit between the two proteins. So that's, this is the news that I would like to share with you regarding the function of the protein in the inner mitochondrial membrane. But then, I mean, if you have a look at the literature, once the protein is translocated into the cytosol, the interacting mapping is limited to the apoptoson formation and to interacting with members of Bax and Bax protein. So we were so surprised that a protein that is able to is very promiscuous in the sense that it's able to interact with so many targets. Once the apoptosis is triggered, the interaction mapping is limited to only to two partners. So we decided a long time ago to do some proteomic approach using affinity chromatography and mass spectrometry to identify potential human cytochrome C targets. And we identify actually 24, but then you need to corroborate because you have a lot of false positives in proteomic studies. So we corroborate in vitro using biophysics and also in cellular biology. But still, we were able to take a few of them, which are the ones that are in the middle of the slide. And coming back to the idea of transient life versus stable death, what I want to remark with this slide is that this kind of non-redox new complexes are still very, very stable. So the lifetime of these complexes is comparable with the one that cytochrome C is performing with APAF1. So still, all the interactions that extramitochondrial cytochrome C is performing are characterized by high affinity. But again, all the residues are surrounding the heme group independently if it is a redox process or not. So this is now the interacting map that we have defined for the extramitochondrial cytochrome C in the cytoplasm and in the nucleus. So let me just focus first in the cytoplasm. So as you know, because this is in the textbooks, extramitochondrial cytochrome C is able to interact with APAF1 to form the apoptoson and to activate the cascade. This is the apoptotic intrinsic signaling pathway, the typical one. But we have identified other partners, like, for example, this chaperone, 1433 epsilon. By the time that we discovered that this chaperone was a partner of cytochrome C, it was also published that this chaperone was able to inhibit APAF1. So to have a look in detail this signaling pathway, we know that cytochrome C interact with APAF1 at the level of the double D40 domains of APAF1, and this is quite important to understand the following experiment. And this complex is able to interact with caspase-9 to form the apoptoson and to activate caspase-3. On the other hand, we know that APAF1, and more specifically phosphorylated APAF1, is able to recognize 1433 epsilon to form this kind of complex and to block the apoptoson formation. So the question is, what is the role of cytochrome C, of the extramitochondrial cytochrome C, not in this well-defined signaling pathway, but in the other one? So we decided to check if the two proteins, if the chaperone, 1433 epsilon, and cytochrome C, were able to interact each other at the endogenous level, so without overexpression. And we test that indeed they are able to interact only when we induce DNA damage using apoptotic. Apoptotic is a kind of DNA damage inducer that inhibits one of the topoisomerase. So we were able to check the interaction by immunoprecipitation, and the interaction occurs in the cytosol. Then we did some NMR, some ITC, and also some mutation to define the possible structural model between the chaperone and the heme protein. I didn't tell you the chaperone is an homodimer. It has this shape like a cup with a concave groove and a convex groove, and cytochrome C was able to explore both sides of the chaperone. So the stoichiometry is 2:1, 2 molecules of cytochrome C with 1 molecule of the chaperone, but still we didn't know what was happening. So we performed some caspase-9 activity assays using a truncated form of APAF1. So a form of APAF1 devoid of the double D40 domains. In other words, this construct was not able to recognize cytochrome C, but still was able to induce caspase-9 activation. And still it was inhibited by the chaperone, by 1433 epsilon. So when we incubate the three proteins with increasing concentration of cytochrome C, we recover the activity, which means that cytochrome C is sequestering the population of the chaperone, and then the chaperone cannot interact anymore with APAF1. And if we move to the phosphorimetic mutant of APAF1, because it was discovered the residue that became phosphorylated, in this case was a serine, we observe more inhibition, because the phosphorylated one is more appropriate to interact with 1433, but still we recover partially the activity of caspase-9. So coming back to the signaling pathway, what we think that is happening is that cytochrome C, the pool of extra mitochondrial cytochrome C in the cytosol, because it's huge, the amount that is released into the cytosol, is able to sequester the chaperone and then speeds up the caspase-3 activation through the canonical signaling pathway. So it's a new function for a protein in a different compartment. It's not only involved in interacting with APAF1, but also inhibiting the role of the chaperone. But let me just move to the nucleus, because this was quite surprising for us when we identify new targets of cytochrome C in the nucleus upon DNA damage. And the common function for all these proteins, SET, nucleophosphine, and ANP32B, is the fact that they are histone chaperones. And histone chaperone is a group of chaperones that are able to interact with histone to promote the assembling and the disassembling of the nucleosomes. So we decide, first of all, to check if cytochrome C travels from the mitochondria into the nucleus upon DNA damage. So for that, I mean, we use different DNA damage inducers, different cell lines. We check endogenous levels, overexpression of the protein, everything that you can imagine. And I'm not a cellular biologist person. I'm more a structural biologist person. But the reference asks for tons of cellular experiments. So we tested at least as much as I know about the system. But in this case, what I show you is just a cell line that constitutively expresses a chimera between cytochrome C and GFP. So in principle, in control cells, so without any stimuli, the protein is mainly in the mitochondria. And you can use MitoTracker to check that this punctated fluorescence corresponds to mitochondria. But upon DNA damage inducer, we observe the fluorescence not only in the mitochondria, but spread out through the whole cell. We did obviously some cell fractioning, and as you can see, even after 1 hour CPT treatment, the protein was either in the membrane, in the mitochondria, or in the nucleus. But we didn't observe so much. There is a bit amount of protein in the cytoplasm, but there is not so much protein in the cytoplasm. Of course, as I told you, we tested not only CPT, we tested all other DNA damage inducers, and we checked that we were producing the DNA damage just monitoring the levels of this gamma H2XA, which is a DNA damage hallmark. And even more interesting was the fact that when we observe what is happening at the level of the cytoplasm, so when we observe the activation of caspase-3, we didn't observe activation of caspases. So Cytochrome C is translocated into the nucleus without apparently passing through the cytoplasm and without activating caspase 3. And this is not the case when we use other apoptotic inducers through oxidative stress through the mitochondria and not through the nucleus, because then we observe the activation of caspases. It is very nice when other groups corroborate your finding, and this is what's happening with the group of Nur Kamal and co-workers, because he observed that the protein was in the mitochondria and also in the nucleus upon CPT treatment and upon UV treatment. But it's even more interesting, this transmission microscopy data that a group in France published, in which they observe nuclear cytochrome C before the activation of caspase 3. So only when cytochrome C is in the cytoplasm, then caspases are activated. So with this in mind, we decide to explore what is the role of cytochrome C in the nucleus. So we start studying the interaction between the heme protein in the nucleus with the three histone chaperones, with NMP32B, with Z-TAF1-beta, and with nucleophosphine. Because we start thinking that probably it has a role in the DNA damage response. And the DNA damage response has different levels. So we need to signaling the DNA damage, and in this signaling step, we think that it's important interaction with NMP32B. We need to repair the DNA damage if the DNA damage is not so high. So, I mean, we need to give some time to the DNA repair machinery to access to the naked DNA and to repair the damage. And finally, what we call execution, in which nucleophosphine is involved. As you can see, I mean, they are, from the structural point of view, they are quite different. This is a monomer, this is an homodimer, and this is a homopentamer. But what is interesting is that they have a globular domain, but they have also intrinsically disordered domains at the C-terminal end. So in the case of NMP32B and Z, this intrinsically disordered domain is rich in acidic residues, in aspartate and glutamate, whereas in the case of nucleophosphine, this intrinsically disordered domain are between the N-terminal domain and the DNA binding domain, and they are stretches of acidic and basic regions. So let me just start with the interaction between NMP32B and cytochrome C in the context of signaling. So I told you before that the way to check that you are inducing DNA damage is monitoring the gamma H2AX signal. So the first thing that we think was to check the levels for these histones in wild-type cells and in knockout cells in cytochrome C. And the levels are really different. So this histone start signaling the DNA damage, and then there is a phosphatate, which is called PP2A, that dephosphorylates the histone to continue to the repair of the DNA damage. This is the process. And this phosphatate is inhibited by NMP32B, which I told you is a target for cytochrome C. So we decide, okay, let's check if under DNA damage, both proteins, NMP32B and cytochrome C, co-localize in the nucleus, and indeed, if the two proteins interact in cell. And this is a technique that you probably have heard. It's the proximity legal assay that allows you to determine interaction between NMP32B and cytochrome C in the cell. So the advantage is that you can monitor in which compartment the interaction is happening. And it's measured by these red spots. So under DNA damage conditions, you can see more red spots in this figure. And if you quantify, it's clearly that when cytochrome C is in the nucleus, it's able to interact with NMP32B. And we corroborate this data also by pull down. So then we say, okay, well, which part of NMP32B is responsible for the interaction with cytochrome C? So let me just give you a brief summary of the domain organization of this protein. It contains this globular domain and this disorder C-terminal domain. And as I told you, it's an inhibitor of the PP2A. So we monitor PP2A activity in the absence and in the presence of NMP32B, and as you can see, it's partially inhibited. But when we incubate with full-length protein and with increasing amount of cytochrome C, we recover partially the activity of PP2A. And this is not the case when we work with the N-terminal domain. So the data tell us that probably cytochrome C is recognizing the C-terminal domain, and this is the reason why it's changing probably the dynamics of the N-terminal domain, and then it has a consequence in the PP2A activity. But we corroborate this by NMR, so this is just an example of one signal, of one amide signal upon titrating with the full length in comparison with the N-terminal domain of the Histone Chaperone and also with ITC. So we were quite confident that the C-terminal domain is the place that cytochrome C explores in NMP32B. The following question is what is happening with the N-terminal domain. And as you can see, the N-terminal domain is this is a docking with restrictions that we get from a paper that was published between PP2A and NMP32B. But it looks like the N-terminal is in contact with the catalytic and the regulatory subunit of PP2A. And actually, if we have a look in detail, the catalytic center of the phosphatase, it's composed by two manganese, which are very close to a side chain that is aspartate 31 in the Histone Chaperone. Okay? So we decide, okay, well, what is happening here? We did some molecular dynamics because we couldn't record NMR data on the full protein, and actually now the assignment is down by not in 32B, but in 32A by the group of Martin Blackledge. So now we can monitor what is happening experimentally with the LCAR, but we know by ITC and by NMR that is the LCAR, the disorder, the CN disorder domain, which is called LCAR in NMP32B, that is in contact with the heme protein in a stoichiometry of 2 to 1, 2 molecules of cytochrome C per molecule of the Histone Chaperone. And what is happening is when cytochrome C is bound to the disorder region, the dynamics of the N-terminal domain drastically change. So it looks like it is more dynamic. So this is what we call long-range allosteric effects by the binding of cytochrome C. We quantify the amplitude of these movements and also the direction of these movements, and you can easily clear that the amplitude of the movements in the free protein is significantly different with respect to the complex. And actually, if we have a look at the side chain for the aspartate 31, which is close to the catalytic side of the PP2A, the movement is also very different. I will not show you, but we have data of the mutant of this aspartate 31A alanine mutant, and indeed it behaves as the complex with cytochrome C. So everything, it makes sense. So just to show you a model of all the data, what we think that is happening is under homeostasis. We have an NMP32B acting as an inhibitor of the PP2A, so then the DNA damage signaling is silenced because you don't need it, we are under homeostasis. But when the DNA damage occurs, then the heme protein travels from the mitochondria into the nucleus. It binds the C disorder domain of the NMP32B. This has an effect in the dynamics of the globular domain and promote the activation of PP2A. So then PP2A can dephosphorylate the histone that is signaling the DNA damage. So this is in terms of the first complex, and for the second complex, for the Z-TAF1 beta, I would like to show you that not only the C terminal domain, but also the globular domain, what we call here ERMAF, are really, really flexible in this protein. And this is, well, not a problem, but a kind of issue when you want to work with this domain together with the heme protein. And actually, we have some data, SAX data, sorry, that we need to include motions to fit properly the data. But to explore which part of this globular domain is the one that cytochrome C used to interact, we use EPR, PRE-NMR, and fluorine-NMR. So we decide to mutate several residues in the globular domain of Z-TAF1 beta, so in the dimerization region, and also in the interface between the ER mass plus the residue in which the LCAR, the disorder domain, starts. So we attach these cysteines to a paramagnetic probe, and we measure the EPR spectra with and without cytochrome C, and also we observe the effect in the intensity of the amide group of the reduced cytochrome C. And in parallel, these cysteines were attached to a fluorine probe to monitor 1D fluorine NMR, to see if we can monitor chemical shift due to the binding with cytochrome C. So these are the data for one. I will show you just two positions. The one that is suggesting that is exploring by cytochrome C, and the other one that does not. So if we attach the probe in the ER-MAF, in the interface between this ER-MAF, we see a kind of broadening in the EPR. We monitor some shift in fluorine, and we observe some PREs on the surface for amide of cytochrome C. That when we plot on the surface of cytochrome C, you can see that the residues that are more affected are surrounding the heme. And on the other hand, when we label the dimerization helixes, we didn't observe any kind of fluorine NMR chemical shift, not broadening in the EPR, but also negligible PRE effects. So we can conclude that the dimerization domains are not explored by cytochrome C. So we built an ensemble of conformations of cytochrome C, just exploring the interface between the ER-MAF. What is happening with the LCAR, we don't know yet. We are still working with the intrinsically disordered domains. Again, that's fine, this is the structure, but what is the consequence from the functional point of view? Well, we start thinking that maybe cytochrome C is able to compete with the histones for the binding site on set, and to test that, we did some electrophoretic mobility shift assay. So you can see here the band for the complex between histone and set, and the band for the complex between cytochrome C and set. And if we increase concentration, if we increase the amount of cytochrome C, we are moving from one position to another. So it suggests that the heme protein is able to compete with histones for the binding site in set. And we did that also by NMR, because working with metalloproteins sometimes is not nice, but from time to time we can use a really well-separated signal, and this is the case for reduced cytochrome C. This is the signal of the methyl group of methionine 80, which is the axial ligand of the iron. And we monitor this signal for free cytochrome C for the complex with the chaperone, with the histone chaperone. It's broadening beyond the detection limit because of the large molecular weight. And when we incubate with H2B, H3, or H4, we recover the signal. So it suggests that, again, I mean, there is competition between the histones, the isolated histones, and cytochrome C. But still, we don't have evidence about the function. So I told you that these histone chaperones are able to interact with histone and to promote a kind of supercoil in the plasmid. And the good thing is that the way that relaxed plasmid and supercoil plasmid is different in a gel. So we can easily distinguish the relaxed plasmid with the different supercoil state for the plasmid. So when we incubate the chaperone, the histone chaperone with the histones, we observe a kind of supercoil state for the plasmid, but when we include in the equation cytochrome C, we lose this supercoiling. So we think that once cytochrome C is in the nucleus, it competes with the histones, and to keep the DNA naked, to be repaired by the DNA repair machinery. I don't have data about the DNA repair machinery, but we are working on testing in collaboration with a group in genome dynamics about on testing if the DNA repair machinery is more efficient when cytochrome C is in the nucleus. And finally, our favorite protein, and this is a nice collaboration with a group of Juan Hermoso here in the Roca Solano. We decide to explore the interaction between nucleophosmin and cytochrome C. So Let me just show you very briefly what was the problem that we're trying to answer. So nucleophosmin is one of the main components of the nucleolus. And the nucleolus is a membraneless organelle. So it's a kind of structure that is formed inside of the nucleus. So under a stress situation, NMP is attached to ARF, which is the most well-known target for NMP. And then, if this complex is formed in the nucleolus, HDM2 is able to interact with P53. P53 becomes ubiquitinated, and this is a signal for the proteasome. So we keep the levels of P53 very, very low under stress conditions. But upon DNA damage, it was known that ARF and NMP are dissociated, and both proteins leave the nucleolus to be in the nucleus. And then if ARF is free of NMP, it's able to interact with HDM2, and then P53 is not anymore ubiquitinated, instead is phosphorylated, and it means that then P53 is stable, so it can regulate gene expression. And depending on the DNA damage levels, P53 can activate the transcription of genes that are related with the cell cycle arrest, trying to repair the DNA damage, or if the DNA damage level is so high, then P53 just goes into the cytoplasm, open the pore in the outer mitochondrial membrane, and apoptosis starts. So our question was, okay, we know that cytochrome C is in the nucleus. We know that NMP is a target for cytochrome C. Is cytochrome C regulating somehow this signaling pathway? So just let me explain you what we are looking here. We are checking the endogenous levels of the protein. So this is immunofluorescent. This is not a chimerax or protein attached to the fluorophore. And we are monitoring in green endogenous cytochrome C, and in red NMP. So you can see that upon CPT treatment, so inducing DNA damage, there was a co-localization between cytochrome C and NMP in the nucleolus. So cytochrome C is translocated into the nucleus and even in the nucleolus. And we tested the interaction by PLA, as I showed you before, but also by immunoprecipitation, just checking the nucleolar fraction and the interaction between the two proteins. So we were quite confident that the endogenous proteins, upon DNA damage, were able to interact in the nucleolus. The question is, which domain of NMP is responsible for the interaction with cytochrome C? So NMP is an homopentamer. It has an N-terminal domain involved in oligomerization state. It has also a C-terminal domain that is involved in DNA binding. And in between, there is this histone binding domain, which are intrinsically disordered stretches of acidic, as happening in NMP32B and in Z, but also formed by basic residues. So the structure of the N-terminal domain was solved by crystallography. The only problem with the structure was that the loops between the beta strands were not solved, probably because they are very dynamic for the free protein. The structure for the DNA binding domain, the C-terminal DNA binding domain, was also solved. And let me just show you that regarding ARF, which is the target of NMP, the part of the protein that is interacting with NMP was the N-terminal domain that is rich in arginines. And this is just a model because we are not very confident if it's the stretch adopts alpha helix conformation. Well, we tested which domain of NMP was responsible of interaction of cytochrome C, and we did so many constructs, many mutants by ITC, by NMR, and we conclude that it's the oligomerization domain, the one that is responsible for the binding of cytochrome C. We observe binding also with the construct containing the disordered regions and the DNA binding domain, and very little binding, very weak binding with the C-terminal domain. So then we asked the group of Juan Hermoso for help, because this is huge for NMR, so we couldn't afford any kind of structure by NMR, and we asked to solve the structure between cytochrome C and the oligomerization N-terminal domain. And what they conclude is that they were able to see the loop that were not visible in the absence of cytochrome C, and we thought that maybe it's because cytochrome C is fixing the dynamics of this loop, but we were not able to identify any electron density for cytochrome C. And that was really a pain because we needed to confirm that cytochrome C was in the crystal. The crystal was red. The protein, the heme protein, is red. So the crystal was red. We washed the crystal. We tested by Western blot, and also we record the iron fluorescent scans at the bin line to be sure that the iron was inside the crystal. But because we were not able to define the position of cytochrome C, we moved to Valpuesta group, asking for help and asking for at least low resolution electron microscopy. And this is the low resolution structure for the NMP alone. We dug the pentameric core and the DNA binding domain, and when we include cytochrome C, there was a drastic conformational change from the extended, what we call extended structure, to the compact structure because of the binding of cytochrome C in the middle of the core. So cytochrome C is interacting with the N-terminal domain, but there are a change in this flexible and disordered region. Again, we asked, okay, then cytochrome C is able to compete with ARF for the binding site of NMP, and we did exactly the same that I explained you for the histone. So we monitor the signal of the methionine 80 for cytochrome C, and indeed there was a competition, and also we record some EMSA. And if you have a look, those residues on NMP that are responsible for the interaction with cytochrome C and with ARF, all these residues are located in the very negatively charged phase of NMP. So it can be a kind of competition between ARF and cytochrome C. But remember that from the structural point of view, they are completely different. The only common feature is that ARF is rich in arginines, cytochrome C is very rich in lysines. So we still wanted to see if in the cellular context, at least in nucleoli fraction, we are able to see this kind of competition. So we label NMP with, this is with nucleoli in vitro, so we incubate this nucleoli with NMP labeled in green, with a fluorophore in green, ARF in red, and cytochrome C in blue, sorry. And as you can see, by time, cytochrome C was shifting out ARF from the nucleoli. But again, this is in vitro because if you talk to cellular biologists, they don't like even cellular compartment. So we move to the cell to do some FRAP, and as you can see here, we tested the fluorescent recovery when we photo-bleach. This is the nucleolus. So we photo-bleach the nucleolus, and we monitor the kinetics of recovering for ARF in the absence and in the presence of cytochrome C. And you see that the kinetics tell us that the mobility of ARF in and out the nucleolus is significantly increased when cytochrome C is in the nucleolus. So with all this data in mind, we built a model in which under homeostasis, we have an NMP and ARF in the nucleolus, forming a complex that we don't know the structure, but upon DNA damage induced, cytochrome C is traveling into the nucleus. It's able to shift ARF out of the nucleus and to form a complex which is different from at least from the conformation of free NMP. So in summary, I hope I convinced you about a protein that is traveling between compartments and doing different things depending on the cellular compartment in which the protein is. So in mitochondria, the most exciting and provocative proposal that we are doing is the long-range electron transfer involving cytochrome C. And this long-range electron transfer can be modulated by phosphorylation of one of the components, in this case of cytochrome C. In the cytoplasm, it's able to interact with 14-3 epsilon with a chaperone to speed up the cascade activation. In the nucleus, at the level of NMP32B, is promoting the inhibition, the NMP32B dependent inhibition of PP2A, inducing long distant allosteric changes. In the nucleus, it's also able to block the histone binding domains of Z-TAF1 beta, so it's capable to inhibit the nucleosome assembly activity and chromatin dynamic. If this has an impact on the DNA machinery, on the DNA repair machinery, we need to test it. And in the nucleus, we think that cytochrome C interact with NMP, involving the extended to compact transition of NMP and the release of NMP sequestering ARF. And I would like to remark that all this interaction involves several lysine residues on the protein, and that's why we are doing an extra effort in assigning the amine groups of these lysines, because we think that these lysine residues plays a pleiotropic role in the trafficking, in controlling, the trafficking and the availability of proteins in the nucleolus. That's our main hypothesis. So just in the cellular context, what we propose is that the protein as p53, so it's not so strange, plays a dual regulatory role in cell fate decisions. First as a protector in the nucleus, in the dam of the DNA damage response by altering chromatin dynamics, the interaction with Z, controlling p53-mediated cell cycle arrest, the interaction with NMP, and recovering PP2A activity to facilitate DNA repair and let the cell live, the interaction with NMP32B. But if the level of the DNA damage level is so high, then the protein, then cytochrome C, is massively translocated into the cytoplasm, and then it's able to activate cascade activation and to lead the protein into apoptosis. And with that, I hope you come with me in the journal between cellular compartments, and thanks very much for your attention. Thank you.
So thank you very much, Irene, for this excellent talk. So I think it's time for questions. Those attending by YouTube, please write your questions in the chat. So please, your questions. Yes, Armando.
Thank you very much for your talk. For me, it's surprising that that amount of different activities outside of the mitochondria, which are not related to the redox properties of the protein. Mm-hmm. So can you comment on that?
I mean, they are not dependent because we have tested both redox state of cytochrome C, and they are not dependent in terms of binding. Yeah, I mean, because we think, and this is just speculation, we think that it's not depending on the heme group, it's depending on the lysine stretches that we think, that we explore. So it's not a question of transferring electron, it's a question of multivariant that cytochrome C offers to interact with the different histone chaperones and other partners. So yeah, the first thing that we did is testing if they were depending on the redox state. They are not depending on the redox state. They are not, involving electron transfer. We think it's more depending on the charge, the number of charge and the sequence of the charge that the protein has. And and we are also, and this is just, totally speculation, because one thing that we are not totally sure is the fact that the protein that is translocated is the folded one, not even the redox one, okay? So we start thinking that it behaves like a polylysine change. And that's why there is a lot of amount of protein of cytochrome C synthesized. But this is, again, this is totally speculation. We are now exploring the levels of the protein that is newly synthesized upon DNA damage, if it is new, if it is the bulk that we have in the cytochrome. Okay, so, but yeah, that's because of this lysine. And if you explore the kind of interactions, it's true that it's not the same interaction for the arginine. I fully agree. But still, lysines can promote liquid-liquid phase separation, and we start thinking that is probably because the protein is not folded. Yeah, so we will see. Yeah.
Thank you for this fantastic and very, very, what David would say, lots of chicha, so much results.
A lot of
fantastic.
teaching. Yeah. A lot of teaching. Yeah, that's true.
I was wondering, you mentioned that in the mitochondria, the interactions tend to be very transient as it goes between the complexes 3 and 4, and then in the cytoplasm and the nucleus, they tend to be much more stable. But there's one interaction I saw with ANP32B, where you showed the NMR spectrum, where it seems like you have some intermediate exchange there, which is usually more typical of a weaker interaction.
Well, intermediate exchange is weaker, I agree with you.
Or fast exchange probably is.
Yeah. Well, I mean, it's in the range of micromolar, the KD. Okay? In the range of 5, I cannot tell you exactly the number because I don't remember, but it's 5, 10 micromolar. But the K-off, and we measure in interferometry, is rather long. Okay? That's why,
interesting case.
yeah. And the story with an NP32B now is totally open because if we can't have a look at the LCAR, I mean, we will do it for sure to do the PREs and so on. Then we will discover if it is really fast exchange, yeah. But when we talk about transient complexes in electron transfer, I mean, the KD is around 100 micromolar normally. And it's very, I mean, the turnover is very, very fast. It's different from the binding, yeah.
Thank you.
Thank you very much.
Thank you, Douglas. Yeah, Javier.
Thank you. Can you hear me? Thank you, Irene, for this. Very nice to see you. Thank you for this fantastic talk. As Doug mentioned, massive amount of data there. I was just wondering if this protein, cytochrome C, is involved, as it was asked previously, in so many different functions. I guess that it will be largely modified. And if this were true, how would this affect interactions that you were showing us here, considering that these regions of particularly the chaperones act sort of like dipoles, no?
That's a very, very good question. Indeed, it is tightly regulated, and what we think is that, for example, the acetylation of lysines is going to be very, very important in the interaction with the functions in the nucleus, but we didn't have time to explore it. I mean, we are doing some ITCs to see if we can check the KD. But my feeling is that depending if the lysines are acetylated or not, the interactions with the disordered regions of the chaperones are going to be completely different. And it has been described that it can be acetylated in different positions related with several illnesses and so on. Yeah, it's a fantastic question. But I need another 10 years. And also funding. That's going to be great. Yeah. But not only the PTMs, not only the post-translational modification, the crucial question here is how the protein travels from the mitochondria into the nucleus without passing the cytoplasm. Because the mitochondria is fine, I didn't tell you, but we check the mitochondria, and when the protein is in the nucleus, the mitochondria is perfectly okay, because there are a huge amount of protein. The protein is synthesized in millimolar concentration. So you have a lot of protein for transferring electrons between two complexes. So this protein must be something, must do something, yeah. So how is traveling? So we know now that there are cross communication between organelles, and there is a path that was a group in UK. They discovered a path between the mitochondria and the nucleus. So we want to check if cytochrome C is traveling through this channel, and then you don't need to activate caspases, which is the tricky point here. Yeah.
Okay, some more questions. I think there is no questions in the chat. I am very surprised to see that you mentioned that the distance at which electron transfer between two redox partners can occur can be up to 10 nanometers. This is huge. So how this distance physical distance, combined with the fact that in the biological environment is so crowded. What does it mean?
I mean, we think that, but again, I mean, this is a fantasy. I mean, we are now, that was a collaboration with Pau, and at the beginning it was like, okay, let's check the distance. Okay, we will try. And then it came up that it is huge, as you said. And it's not only for this system. It's happening also in photosynthesis. We have already tested in photosynthesis. And also it's very dependent, as I told you, on the charge. So what is the relevance in the context, in the cellular context? What we think is that you don't need to be close enough to the membrane complex, 3 or 4, and you don't need to have a defined orientation. So you can transfer electrons in a different way, and this is plasticity in some way. So we think that the idea that we have that you need, to be close and to exclude water molecules and to is not what is happening in the cell. And this is more efficient, actually, in terms of electron. Actually, we measure the electron transfer, and it's faster in this context. How the crowding environment affect that? I don't know. I mean we should measure in cell, but I don't know.
It is possible that other potential aberrant transfer can occur in this case.
You mean to the other partners that you have? Yeah, but I think it's, I don't know the KT, the electron transfer kinetics constants for the different complexes. I don't know if they are equivalent. I don't know if this is optimized for the mitochondrial electron transfer. But yeah, in principle, yeah, it can be, definitely. Yeah, well, I mean, I can tell you a story. I was discussing with Pau, and we are asking for some grants, and the question from the person that is thinking to give the money was, okay, can you measure in cell? I said, not me, no with me, This is yeah, well, we don't know. But still, I think that you can have a lot of biotechnology applications, biosensors that then you don't need, for not thinking in cell, but thinking in, sorry. Me lo he cargado, seguro que me lo he cargado. Pero puede ser,
Okay, so yeah, another just to finish, just one general question I would like to comment with you is that you have discovered somehow a new function for cytochrome C, but you are analyzing your systems in a interatomic level.
Yeah.
So which is function for a particular protein in a biointeractomic world? I don't know if my question, do you understand my question? I mean, if you are analyzing your system as a network with many partners, in which is the network the relevant unit, functional unit, so which is the function for a protein of just one member of this highly complex network?
You mean to the partner? What is the function of the partner?
Of, in this case, cytochrome C.
Of cytochrome C in all these
in all this network.
Well, I think it's dependent on the stress conditions that you have. I mean, if the cell is fine, is under stress condition, then I mean it's in the mitochondria transferring electron. If you have a DNA damage, then there must be a signal coming from the nucleus to the mitochondria that allow the protein goes. But still, the mitochondria is function, so you can working with the cell, and then in the nucleus, and this is what we are doing now, is the protein try to repair, not directly, but keeping the DNA accessible for the repair machinery. So these two functions can be at the same time. Yeah. Okay, but if the cell, if the protein is in the cytoplasm, no way. I mean, the cell is going to die. So the mitochondria, I don't know if I answered your question.
No.
Okay.
Probably my question is not well done.
Okay.
We will talk later if you want. So just to finish, let me give you a small present for your participation here with us. So thank you very much, Irene.
Thank you
Yes,
so
of
much.
course. Thank you.
Oh, it's a cup. Thank you very much. Perdonare, che